Hello everyone, my name is Jiacheng. Today I'm going to have a talk about single molecule for spectroscopy to study instant titling that are likely causing cardiomyopathy. So what is titling? Titling is a very large protein that can be found in human muscle, in particular our heart muscles. As you can see in that figure, the red line is the titling. It connects the the Z disk and the M line. It's really big, contains more than 300 domains. So just because it's really big, some mutations always happen in titling. And in some previous study, people sequenced more than 800 variants, and they use bioinformatics analysis and the consegregation analysis. They classified the 800 variants into three kind of group: the likely variants, possibly, and unlikely variants. And in their study, they pay more attention to the possibly and likely variants, and they, as you can see in the figure, they labeled the specific exon with possibly and likely mutations. And they divide the titling into three parts: the Z disk, I band region, and A band region. So, but in our study, we pay more attention to the I band region because the titling plays a very important role in the passive tension and the elasticity of our muscle, and which the I band takes this kind of responsibility. So here's the schematic of I band. So it has more than 100 immunoglobulin-like domain, which we call IG domain. So here are 7 mutations that are likely causing the cardiomyopathy, and we pay more attention to those 2 likely mutations, which is the I-84 and I-94. So, but the first problem we had is we don't have the crystal structure of the protein, so then we use the homology modeling to model the protein structure. So as you can see, here's the result. We got 4 protein, and as you can see, all the protein has the similar beta sandwich structure, which is similar as the IG domain in some previous study. And as you can see, they have similar structure. Then here's a question that are they have similar stability in thermodynamics and mechanics. So to study the thermodynamics stability of the protein domain, we use the chemical induced denaturation. So typically, we take the tryptophan as a probe to study the stability, because when the tryptophan was buried in the protein, it can be have a high intensity inflorescence. But when the protein was unfolded because of some denaturant in different concentration, then the fluorescence will change, and in that way we can get the fluorescence fraction, the folded fraction in different concentration of denaturant. Then here's the result. As you can see, for example, I-94, sorry, we can directly see a shift to the left in the curve, and which gives us the delta G, the free energy, 1.2 kilocalories per mole. And the same result is also found in I-84. It also gave us a delta G, which is about 0.9 kilocalories per mole. For both of the results indicate that those 2 mutations lower the thermodynamic stability of either I-94 or the I-84 domains. And to study the mechanical stability of the protein domain, we use single molecule for spectroscopy. So typically we just incubate the protein on a glass cover slip, and we have an AFM cantilever that gets approached to the surface and pick up a protein. And as you can see, the figure B, when one protein was unfolded, then we can see a peak, which is shown in the figure C. A peak indicates unfolding event in the force extension curve. And finally, we will do the warm lecture model to fit the curve, which is the red dot line. We get the distance between two warm lecture fit, and that's what we call contouring increment. It's kind of like a fingerprint or ID of the protein domain, because like most of the protein have its own contouring increment, and to make the experiment in a high efficiency, we used some specific labeling to do the FM experiment. So in some previous study, people has already found a specific interaction, which is the X-dot and cohesion interaction. They can recognize each other and form a complex. They have very high affinity, high rupture force. The rupture force can reach to about 500 to 600 picnewtons. And in the study, we modify the X-dot protein onto the cantilever and the recombinant protein cohesion and the GB1 Ig domain tetramer onto the surface. So, and it's important to know that the GB1 we used in the study is to be a fingerprint for our protein. Because it's a well-studied protein, we used it to help us recognize, oh, that's our protein, because it has several peaks with contouring increment of 18 nanometers. And here's the result. As you can see, we have some typical curves here. In total, we have 8 domains, which means we're going to get 8 peaks in the contouring in the force extension curves. And as you can see, there are 4 of 18 nanometers peaks, and the rest of the peaks must be our protein, our target protein, which is the I94. And then we collect the I94 unfolding force and also the mutant protein. And finally, we made a histogram and get the average force. So for the wild-type protein of I94, the average force, unfolding force is 187 picnewton, and when it's mutated, it's 145 picnewton. So it's decreased dramatically. And also we do the same experiment at different pulling speed. As you can see, we see the figure below here. And we feed the curve with Monte Carlo simulation, and then we can get the force distance and the information of the unfolding rate constant. And we found that the mutation accelerate the unfolding rate, and to drive a kind of conclusion, the mutation can lead a strong destabilization in mechanical stability of the I94 domains. However, things are a little bit different found in I84. As you can see, once we got the histogram of the unfolding force, then we find that the wild-type protein and the mutant have similar unfolding force. It's just 10 picnewton in difference. And also we did the pulling speed experiment, and finally we can see a slight accelerate of unfolding rate for that protein domain. And we have to say that those kind of mutations lead to a modest destabilization in mechanical stability. And finally, we did a folding experiment to get some folding constant data. So typically what we did is pick up a protein, but we didn't break up the specific interaction, that we can release it and stretch it again. So then we will change the relaxation time. So as you can see, from when the time relaxation time is about 5 milliseconds, most of the red peaks, which is the I-94, cannot be refolded. But if we increase the relaxation time, you can see most of the protein was refolded. We do the same thing at different relaxation time, and finally we can get a refolding probability versus the relaxation time plot, and we fit it with first exponential function. Then finally we can find the folding rate. So as you can see here for I-94, the folding rate was decelerated by the mutation for both of the protein. And finally, to draw a conclusion, the I-94 mutation can have a significant destabilizing effect, both thermodynamic and mechanic on I-94 protein domain. However, for the I-84 mutant, the mutation lead to really modest destabilization of I-84, because it only have a reduction of unfolding force in just 10 piconewton, and this result will be just the first step. We trying to understand the mechanophenotype of cardiomyopathy causing mutations in the elastic I band, and we are trying to do more research about some other like Ig domain protein mutations that are related to cardiomyopathy in the future. And finally, I would like to just thank the co-author of this study, Denhuang and Jiahao, and also my professor, Hongbin Li, and also the rest of other group members helped me a lot. And finally, I would like to thank Heart and Stroke and NSERC from their fundings. That's all. Thanks for listening. I would like to answer questions.